Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine this scenario. You get bitten by a deadly snake, you get the treatment, but the treatment itself could, well, make you sick.
Mm.
That's kind of been the tightrope we've walked with traditional antivenom.
Yeah, it's a real challenge. But what if we could make that treatment almost 3 times stronger and significantly safer?
That's exactly what we're talking about today. Some really exciting new research coming out of Brazil seems to be pointing in that direction.
Right. We're doing a deep dive into a new, potentially much more effective snake anti-venom serum. It's developed by researchers at the Federal University of São Paulo and the Butantan Institute.
And they're focusing specifically on bites from snakes in the Bothrops genus.
That's right, particularly the Bothrops jararaca. It's a very common and, frankly, dangerous snake in South America.
Okay, so to really get into this, we're looking at a couple of things today. There's a health news article covering the main points, and also the original scientific paper itself. It was published in the Journal of Proteome Research. Bit of a mouthful, but the title is Enhancing the Bothropic Antivenom Through a Reverse Antivenomics Approach.
Yeah, quite a title. But basically, our mission today is to break down how this new serum works, why it's potentially such a big step forward, and, what it could mean for treating snake bites globally.
And we want to do that without getting bogged down in super technical stuff, keep it clear and understandable.
Exactly. So maybe we should start with the basics, how antivenom is usually made.
Good idea. So the traditional method has been around for over a century, right?
Pretty much, it involves injecting small, sublethal doses of snake venom into large animals, often horses.
And the horse's immune system kicks in, starts producing antibodies to fight off those specific toxins in the venom.
Correct. Then, after a while, they collect blood from the horse, process it, purify it somewhat, and you end up with what's called heterologous serum. That's been the standard treatment.
But here's the kicker, and something this new research really highlights. Not everything in that serum is actually doing the work of neutralizing the venom.
Not even close in many cases. Studies have shown that sometimes only about 10%, maybe up to 40% of the traditional serum actually contains those crucial venom-fighting antibodies.
Wow, so a huge chunk of it is just other stuff from the horse's blood?
Essentially, yes. Other proteins and components, and that's where the problems can start. This new study looks specifically at the standard antivenom for Bothrops jararaca.
Okay.
And they found that horse albumin, just one type of protein, made up about 8.6% of that standard serum.
And albumin, while normal for a horse, isn't so great when you inject it into a human.
Right. It can trigger adverse immune reactions. Professor Alexander Tashima, who led this study, pointed out that even with better purification over the years, these reactions still happen. They reported rates anywhere from, 5% up to 57% of cases?
57.
Yeah.
That's really high. And it's often the body reacting to those horse proteins, like the albumin.
Precisely. So the researchers, they set out to figure out exactly what was working in the standard serum. They wanted to quantify the proteins that actually bind to the Bothrops jararaca toxins.
How they do that? Sounds complex.
Well, they used some pretty advanced tools, things like affinity chromatography, surface plasmon resonance, and mass spectrometry, really digging into the molecular details.
Okay, and the big reveal. What did they find in the standard stuff?
They found that only about 27.8%, let's call it just under 28% of the components were actually interacting with the venom toxin.
So less than a third, meaning over 70% was essentially not the active ingredient for fighting the venom.
Exactly. And that obviously presented a clear target for improvement. If you could somehow get rid of most of that other 70+%.
You'd have something much more concentrated and maybe safer too, right?
That was the hypothesis. So they took the standard antivenom and added a new purification step using that affinity chromatography technique we mentioned.
Okay, break that down a bit. Affinity chromatography.
Think of it like molecular fishing. They took the Bothrops jararaca venom toxins and basically stuck them onto a special material in a column. That's the bait.
Got it. Baiting the hook with the venom itself.
Exactly. Then they pass the standard antivenom serum through this column. The antibodies in the serum that are designed to target those specific toxins, the ones they're looking for, they grab onto the toxins stuck on the column, they bind.
Ah, I see. And all the other stuff.
The horse albumin, other non-specific proteins, Most of that stuff doesn't bind strongly, so it just washes right through and gets discarded.
Clever. So you're left with just the specific antibodies stuck to the column.
Right. Then the final step is to release those bound antibodies from the column using a special solution. What comes off is the new improved serum.
Okay, so they made this improved serum. Did they test it to see how much cleaner it was?
Oh yeah, the analysis was pretty striking. They found 87% less albumin compared to the original serum.
87%. That's huge.
It is. And reductions in other non-essential serum proteins ranged from 37% up to 83%, so significantly purer.
Okay, pure is good, but the real question is, does it work better? Did they test its effectiveness?
They did. That's the functional analysis part. Using that surface plasmon resonance technique, they found the new serum had a 2.9 times greater affinity for the venom toxins.
Affinity, meaning it binds more strongly, like a stronger magnet.
Exactly. Almost 3 times stronger attraction to the target toxins.
And what about in like a biological setting? Did they test it in animals?
Yes, they did experiments with mice that were given the venom, and the results there were also very impressive. They found they needed a 2.8 times lower dose of the new serum to effectively neutralize the toxins.
Lower dose needed means it's more potent.
Correct. They measure this as the median effective dose, or ED50. Needing almost 3 times less serum to get the same protective effect is a major improvement in potency.
So let's connect the dots here. We have an improved serum that's significantly more potent.
Requires a smaller dose.
And it has way fewer of those unwanted horse proteins, particularly albumin.
Right, the stuff likely causing many of the side effects.
So the implication is potentially twofold. Smaller doses needed for treatment and a lower risk of adverse reactions for the patient.
That's the hope, and it's a really significant potential advance, especially when you think about the scale of the snakebite problem globally.
Yeah, it's easy to forget just how big an issue this is. The numbers are staggering, over 5 million bites a year.
Yes, leading to over 100,000 deaths and something like 400,000 people left with permanent disabilities annually. It's devastating.
And it disproportionately affects people in rural, often poor communities, young workers, children.
Absolutely. The World Health Organization even reclassified snakebite envenoming as a neglected tropical disease back in 2017, just to highlight the urgency.
So having a treatment that's potentially more effective and safer could make a massive difference in those exact communities.
A truly profound impact, yes, reducing both mortality and long-term disability.
Okay, this all sounds incredibly positive. But, you know, science always has its caveats. We need to talk about the limitations here, right? This isn't available in hospitals tomorrow.
No, absolutely not. That's a crucial point. While the lab results are very promising, this improved serum needs to go through the whole process of clinical trials in humans.
To confirm it's actually safe and effective in people, not just mice.
Exactly. And then there's the whole regulatory approval side of things. That takes time and significant investment.
The researchers also mentioned something specific in the paper about bradykinin potentiating peptides, that the purification might miss antibodies for those.
Ah, yes. That's a technical point, but potentially important. These peptides are another component of the venom, contributing to things like pain and swelling.
Okay.
Because of their chemical nature, the purification process might not capture the antibodies against them as efficiently as it captures antibodies against the major toxins.
So is that a potential downside? Does it matter clinically?
Well, the researchers acknowledge it's an area needing more study. The overall neutralization was clearly much better, but whether slightly underrepresenting antibodies to these specific peptides has a noticeable effect in humans isn't fully known from this study.
And what about the mouse model itself? You mentioned they pre-mixed the venom and antivenom.
Yes, that's a standard approach for initial potency testing, but it's not quite the same as a real bite, where venom spreads through tissue and then the antivenom is administered, often intravenously.
So real-world conditions are more complex.
Definitely. The dynamics are different. Again, that's why human clinical trials are the essential next step to see how it performs under those real-world conditions.
And one more thing, they were upfront, that this study really focused on potency and the composition, right? Not an in-depth safety trial beyond checking efficacy in mice.
Correct. While reducing the problematic protein strongly suggests better safety, dedicated safety and side effect profile studies in animals, and then humans are absolutely necessary before this could be used widely.
It's always good to hear directly from the scientist. Professor Tashima, the coordinator, he stressed how they combined old and new techniques, didn't he?
Yes, he highlighted that blend of classical methods with modern proteomics and other techniques. His goal was clear: Boost the neutralizers, reduce the troublemakers.
resulting in, as he put it, more action even in smaller amounts.
And Tassia Chiarelli, the first author on the paper, she directly linked the increased potency and the lower horse protein levels to that potential for fewer adverse effects.
Which is the big patient benefit, potentially.
Absolutely. Now, looking ahead, this isn't happening in a vacuum. There are other new technologies being explored for snake bites, right? Like monoclonal antibodies.
Yeah, the article mentioned those, similar to some COVID treatments. Very targeted.
Very targeted, yes.
Yeah.
Huge potential there. But the researchers, including Tashima, offered a realistic perspective.
That these new technologies, while exciting, take a long time and are often very expensive to develop and produce.
Exactly. The time, the cost, the clinical trials, it all adds up. So they believe that heterologous serums, like the improved one they've developed, will likely remain crucial, a mainstay of treatment for quite some time yet.
So it's not necessarily about replacing the old way entirely, but about significantly improving it while those other technologies mature.
Precisely. Their work shows that we can make the current standard of care much better using a technique like affinity chromatography. It's about enhancing what we have now, which is vital while we also pursue future innovations.
So maybe a final thought to leave our listeners with. For like 100 years, we've used this anti-venom production method that's kind of like, I don't know, using a shotgun when you need a sniper rifle. Or maybe your toolbox analogy earlier, using the whole toolbox for one specific bolt.
Yeah, captures the idea.
This research shows we can actually zero in on that specific tool, that wrench we need. Makes you wonder, what other long-standing medical treatments out there might benefit from this kind of reverse engineering? Really focusing on isolating the most effective part and minimizing everything else.
That's a fascinating question. It really pushes us to think about how we refine therapies, doesn't it? Not just inventing new things, but optimizing the life-saving tools we already rely on. What does that mean for drug development, for treatment protocols? Lots to think about.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.